It takes one gene to make a protein. There are 150,000 different proteins. How many genes do you need? 150,000. How many genes did we find in the Human Genome Project? 23,000. There's a problem. There are not enough genes to make a human being. So what is wrong is our belief. We are completely wrong about our belief that genes control life. So genes do not control life. The fact. So assumption number 2, false.
Wrong.
Wrong. Good.
I'm learning.
In my research, 40 years ago, I started working with stem cells. People think that stem cells are something new. But I learned very many things from the stem cells. The secret of life I learned from stem cells. Right now, in every one of you, you have stem cells throughout your entire body. And these stem cells can replace any tissue or organ in your body. And you should be able to stay young for hundreds of years, as Greg will talk about. So you might ask, then why do we age? And the answer is because the mind controls the cells and the genes. And we collectively believe in aging. So that my research, early research, I have a tissue culture with multipotential stem cells. These are clones, meaning they're all genetically identical. In my experiment, I take a group of cells, so I take this group of cells and put it into another petri dish with a different environment, environment A. And environment A, the conditions of the medium, the stem cells form muscle. But I could go back and take the very same genetically identical cells and put them into culture dish with environment B, and the cells form bone. Or I could go and take the very same cells and put them in culture dish C with a different environment, and the cells form fat. What controls the fate of the cell? The environment. They were genetically identical, only the environment was different. So I published this work in 1977, and it's a fine structural analysis of normal and modulated cells in myogenic culture. It sounds like Latin, but it sounds like Latin, and so science is like the church, so you can't understand what I'm talking about. But what is the meaning? Is that this is the first scientific report that I made, how the environment controls the fate of your cells. And I use the word modulated, meaning altered, because there was no other word. My colleagues were not interested in this work because they were interested in the human genome. So I left I had tenure, and I walked out of the university as a professor. I left the university because they were not being scientific, because they didn't want to understand this. So I end up at a better university, the Stanford University Medical School. There I repeat the experiments, but more sophisticated. And I get the cover of the journal, Differentiation. But I really wanted the cover of Rolling Stone. But it's science. So after I do this, again, the scientists were not interested. So I left the university for the second time. And I write my book on the biology of how cells work. And in my chapter 2, I lay out the scientific evidence to show you genes do not control biology. I also introduce you to the exciting discoveries of epigenetics, a new field of biology that is unraveling the mysteries of how the environment influences the behavior of cells without changing the genetic code. And I call chapter 2, It's the Environment Stupid. It's a takeoff from President Bill Clinton's campaign saying it's the economy stupid. And 5 months after I write this, people are saying, what do I know? I'm not at a university, so what do I know? And then Nature, in one of the most prestigious journals in the world, Nature, 5 months after my book, an article about stem cells are engaged in a constant crosstalk with their environment. Biologists are fast realizing. No, some of them not too fast. What do you think they call this article in Nature? It's the same story. Science is now saying what I've been saying for 30 years is coming around now, and that there's a new science of how cells work. After lunch, I will show you the science of actually how the cells work, and you will have more knowledge than most doctors in the world today because they still believe in the genes. Now we'll go faster. But this is going to be the fun part because this is where I'm going to explain how the biology actually works. And I will we'll get to the secret of life before we're over. Okay, so what we were talking about is how cells actually work. And it's very similar to this. It's a machine. So just as the gears move and connect and turn and make an action, so do the proteins inside the cell. And there are proteins, I said there are 150,000 different proteins that provide both for our structure and our behavior. So 150,000 different shaped proteins. Now, again, it's like inside the cell, they're just like a clock with all the proteins engaging like gears. So this picture is going to show 3 different examples of proteins. These are molecules blown up very large. And the question is, what makes each protein a different shape? So where does the shape come from? And is underneath the skin, there's a backbone inside each protein, a backbone. And just like a backbone gives you your shape, or you can change your shape, so does a protein. Now, what's really interesting is that all proteins share the same characteristics, so I only have to talk about one protein, but then I'm talking about all proteins. All proteins have a backbone made out of linked units, just like the vertebrae. The backbone pieces are called amino acids. There are 20 different shaped amino acids. And what makes each protein different is the length of the chain and also the sequence of the different amino acids. The DNA, the gene, is the information of which amino acid comes after the other amino acid. But this is not the best model because all the beads have the same shape. So I'm going to use 3 different shaped pipes. A straight pipe, one with a 90-degree bend, and one with a 45-degree bend. That's just 3 out of how many? So, okay, and they're just like the beads, they plug into each other. And these are bonds, and in the real chemical name, they're called peptide bonds. So I will assemble this one protein. And now you can see that the sequence gives a rigid backbone. And one quick question. If I take this apart and put it back together again in a different sequence, will I get the same shape? No. So good, because that means what's the point is that every protein has a unique sequence. So how does it work? Let's go to this picture. Can you in your mind see them moving? Okay, now replace the gears with protein. Okay. Now, in your mind, can you imagine when this protein moves, it moves this protein, and then it moves this protein. And this is a combination of a protein and a metal machine. Now, take away the metal parts, protein machine. Now, there are different groups of proteins. When they move, they make different functions. And the proteins are called pathways. Okay? So there's respiratory pathway, a digestion pathway, muscle contraction pathway. Okay, so inside every cell are protein gears. When they move, they make functions. And most students in medicine or science or complementary medicine study this protein system called the Krebs cycles. Anybody knows the Krebs cycle? And these are protein gears that when they engage create the function of the Krebs cycle. Okay, now are you ready for the secret of